Alex from ExoProTher Medical, and he's a co-founder and a CTO, and he will talk about P53 containing natural nanovesicles of a xenogenic origin as highly effective oncology therapy demonstrated in syngeneic burn models.
Okay, so I'm Alex. As you heard, ExoProTher Medical, we are inspired by evolution. We are developing a breakthrough oncology therapy addressing 60% of human cancers using P53 containing nanovesicles of non-human origin. 60% is because actually in 60% of cancers P53 is mutated. In this talk, I will present you our working hypothesis and the results of application of our therapeutics in vitro and in vivo. So the idea here is a translation of a newly discovered ancient local mechanism of anti-cancer defense into a novel systemic therapy. So now let's time travel 600 million years to the past, to the beginning of the Edicarian period. The period of transition from unicellularity to multicellularity. At this point, the life is, the Earth is prevailed by unicellular organisms, and their life is governed by a basic principle. I will survive and I don't care about the rest. With the transition to the multicellularity, this principle has changed in 180 degrees. Actually, now the life of individual cell is worth almost nothing, and what's important is the prosperity of the whole organism. To ensure this integration of individual cell into the multicellular organism, a new multi-level system of genomic and epigenomic regulation was required. As a result, following control mechanism has developed. Control of proliferation, regulation of differentiation, synchronization of metabolism, contact interaction with neighboring cells and tissues, and of course control of normality. And control of normality is the relevant for our discussion here. So there are 4 types of anti-tumor defense in the organisms. One of them is within the self itself. I will explain every one later. Then originating from surrounding cells, and innate and adaptive immunity. So the first one, within the cell itself, it's based on the mechanism of P53 protein, which is a guardian of the genome. It's responding against the DNA strands and damage. It enters the cell into cell cycle arrest, trying to repair the damage. If it's not working, it enters the cell to the apoptosis. It happens in about 60% of human cancers, and those cancers that P53 is mutated are most aggressive, and they are very resistant to current therapies. The last 2 are widely discussed in the literature as generally accepted theory of immunoediting and development of the classical theory of immunological surveillance. Based on this theories, promising methods of immunotherapy therapy was proposed, and you heard about them in last 2 days. While the last but not least, the second stage of local anti-tumor defense is actually immediate extension of DNA damage repair mechanisms, like based on P53. But in this case, the anti-tumor defense is originating from neighboring cells, and the delivery of tumor suppressors carried through secreted nanovesicles, for example, exosomes, And this is exactly what ExoProTher Medical working on. So a good example of anti-tumor defense originating from neighboring cells is the corneal epithelial cells. Cornea, even if exposed to harmful UV radiation, cancer occurrence is very rare. So being immunoprivileged tissue, meaning there is no blood supply to cornea, corneal epithelial lacks mechanisms of innate and adaptive immunity, and has to fully rely on local anti-oncogenic functions. It is achieved by very high level of P53 expression in the cells, and it's transferred by exosomes to the neighboring cells. So as soon as there is any P53 mutation in one of the cells of the cornea, it gets exosomes with P53 from the surrounding cells. What we can see here, it's electron microscopy of P53 containing exosomes in the tear film of rat. It's happening to all. It's happening actually all vertebrates. We used here MAB 421 as an antibody for P53. And then gold nanoparticles. And here is the same for exosomes. So what do we have actually? Our technology, as I said, inspired by evolution. It originates from tissues where is a very big amount of P53. It's already packed in exosomes. And we harnessing this natural mechanism as a treatment. So ExoProTher, our innovation is actually exosome protein therapeutics. While exosomes gave us non-toxic, non-immunogenic, and flexible routes of administration, protein, we are talking about P53, which is mutated in 60% of cancers. And the mechanism of action is actually selective apoptosis of malignant cells. And we also talking about synthesization of current therapies like radiation and chemotherapy, which are not really helping where there is a P53 mutation, so we can help in this point. So it's a first-in-class tumor-agnostic acellular therapy targeting P53 mutated cancers. We are addressing previous pitfalls of P53 therapeutics. Instead of viral vectors, we use exosomes. Instead of human P53 gene, we use chicken P53 protein, and I will explain why. There is such a thing called dominant negative effect. Till now, all the attempts to use human P53 protein was unsuccessful because actually P53 working as tetramer. So as soon as we provide wild type P53 to P53 mutated cells, there is a tetramization, mutual tetramization between them, and the mutated P53 prevents the work of the wild type. So it's turned out that chicken P53 is different enough to prevent this tetramization, but it's similar enough to perform the anti-cancer function. This is a beginning. Now to some results that we already have. So first of all, here we can see the characterization of our nanoparticles. So ExO2 is a first generation of our therapeutic, while pellet is a kind of purification of nanoparticles could be done using ultracentrifugation or columns. In this case, it's ultracentrifugation, and we have, of course, supernatant. We see the difference in the particles concentration and in the mean side. while here at the end of the day we have particles of about 150 nanometer. When we apply those different fractions on glioblastoma human cells, p53 mutated glioblastoma cells, we see that we have a very high apoptotic rate in purified nanoparticles. And also, if we actually counting the cells, we see that after 24 hours, most of the cells are died. Next, we did the same with murine cell lines. This is specifically MC38, with which p53 mutated colon cancer. And you see the same picture actually. Then we tried to apply our therapeutic, our exosomes on non-cancer cells, and we see there is no reaction. And as compared to the MC colon cancer, actually it's not do anything bad on primary cells. We also have toxicological studies in vivo. Next, we try, first of all, our first generation was exosome that we extracted from chicken corneal epithelial cells. But then we tried to apply the UV radiation on those cells before we extract the exosomes. And what we see that we have a much higher efficacy after UV application. It's supporting the hypothesis of p53 mechanism of action, since p53 is phosphorylated following UV radiation. Next, we tried different cell lines of glioblastoma in vitro. You see three different cell lines here, while two of them are p53 mutated and one of them not. P53 mutated cell lines actually have much higher efficacy, respond very well. And again, it's supporting our hypothesis of p53 involvement here. This is a list of different cell lines that we screened till today. It's lung cancer, it's brain cancer, it's colon cancer, it's skin cancer. All those are human cancers, and some also murine cancers, also colon and glioblastoma. The green ones is the cells that we are working on them in vivo now. I have to say that here cancer with mutant p53 actually responded very well. With the wild type p53, we see very minor response. This is time-lapse video. Can you? Yeah. So this is a treatment. This is a control. This is HT29 cells, the colon cancer, human colon cancer. What we did here, we just two petri dishes with the cancer cells and one treatment. Then we pictured the petri dish for 4 days, one picture per hour, and performed time-lapse video afterwards. And you can see that after 24 hours there is a growth arrest, and then after 2-3 days there is apoptosis, which the time scale is exactly as supposed to be with apoptotic mechanism of action and not toxic. Now to in vivo studies. This was the first pilot that we performed on immunocompetent mice. It's not in nude mice. It was important because our exosomes from different animal origin. So here we injected our treatment for 10 days every day, and we see very clear prolonged survival. Then we go to a little bit different model while we injected our treatment one time in the 3 hours after injection of the tumor. And again, we see pretty clear prolonged survival, while 25% of the animals continue to live for a year or so and die from natural reasons. We did not see any adverse effects, by the way. And the last study that we performed this year, this time as outsourcing, also MC38, urine colon cancer. This time we monitored the tumor volume, and it's very clear that, we got very significant results in the mean of tumor volume. Part of the animals were actually tumor-free. We also saw reduced angiogenesis and increased apoptosis that we performed histopathological findings. Again, no observed effects during the treatment. This is our team. Co-founders, myself and our CEO, Lana Polak, our chief scientist, Evgeny Tendler here, working on P53 about 30 years, is my father. And our R&D lead that now working on scaling of our technology. And we have very important people in our, one of them is Professor Daniel Offen, which is a researcher in the exosome-based therapeutics, is very known. Alexander Panchik, who is molecular biology and immunology. And of course, Avi Travis, who was one of the people that started actually with cell therapeutics in gameta cells. He established gameta cells. That's it. Thank you.
Thank you very much, Alex. Very good talk. Interesting. Any questions?
I'm interested in the nanoparticle aspect. These exosomes, are they stable with time, and what keeps them stable against degradation?
You say stable in time when it's before it's there is injection, after there.
After loading.
We are not loading them. We harnessing them from the cells. It's already with P53 and other actually tumor suppressors. That's the point. We are not developing new nanoparticles. We don't load them.
I see, but they're fully stable with time.
At least we try to freeze them for a year and use it after a year, and it was very effective.
Thank you very much for this interesting talk. Two questions. One, what about the genetic material that still exists in the exosomes while you produce them, and how it affects actually a lot of processes in the body? And the second question, if you can comment on the scalability of the process.
Well, I will start with the scalability. First of all, we work in a number of directions how to scale it, and it can be harnessing of the tissue and then production of the exosomes. That can be also growth of cells in bioreactors. We are working on both directions in parallel, and we will see what will be more efficient. But we don't see any problem here. We are not the first that's growing cells or harnessing cells, so don't see a big deal here. About other proteins, well, there is about 2,000 proteins in corneal cells, right? It's a lot. We're going to characterize part of them. Part of them, in addition to P53, there are another tumor suppressors there that we're going to characterize them. But we don't need to characterize all of them if we don't talking about the regulation. If this was the question.
No, I talked about the genetic material that still exists in exosome. Because they need some of the genetic material of the cells, RNA, so these are things, but RNA. How do you
What we have inside the exosome, we don't try to eliminate it.
It doesn't have any effect.
We don't see any effect in mice. Again, in the immunocompetent mice, we did a toxicity study. We injected, I think, 10 times more than the therapeutic, and we don't see any effect. But I suppose the final say will happen during the phase 1.
Hopefully sooner.
Yes, everybody.
I have a question. How do you estimate the optimal dose, or how do you dose exosomes? By milligrams per kilograms, per particles
per milliliter.
And how do you choose the in vivo dose based on in
We
vitro?
are working on it, you know. we did it here. Already know. Okay. So we took something that we thought that it would be good, and it worked. But of course, we will develop different models and different types of treatment. For example, for glioblastoma, it could be nasal inhalation, and it's completely different formulation. And for lung cancer, it possibly also be kind of inhalation, and it can be injection, intratumoral injection, it can be systemic injection. Everything, all the options are on the table.
Very good. Thank you very much. Thanks again, Alex. Let's welcome Ofra Benny from Hebrew University School of Pharmacy, and Ofra.